Hi students, we are discussing reproduction. In last class, we discussed about sexual reproduction in plants, like flowering plants like datura plants we discussed. So today class we discuss observation of pollen grains and the structure of the oval. Observation of pollen grains. Pollen grain is male parts or female parts. Pollen grain is male parts or female parts. Pollen grain is called as male parts. Pollen grains. So androgen is called as pollen grains. Where we can see in anther, you take any flowers which one having the anthers and put it in the waters, it will float on the waters. You can take this pollen grains and place it on the slide, observe under the microscope. When you observe this in the presence of microscope, you can see the structure of this like pollen grain structure, pollen tubes, and which one having nucleus, nuclei, pollen tubes and nuclei. This is the structure of the pollen grains. Next one is structure of the oval. Structure of the ovals. Where ovals is located in the ovary. In the ovaries. So it's a female part. It's called as gynoecium. Gynoecium, structure of the gynoeciums. So we can see in the embryo sacs there are 7 cells and 8 nucleus present in the embryos. When you look at this structure of this structure of the ovals, you can see inside this ovary, you can see ovals. This embryo sac having 7 cells and 8 nuclei. By the process of mitosis, by the 3 divisions, it will be multiplied and form. So 7 cells and 8 nuclei. 3 times. So the mitotic cell division will take place, and to form a food materials in this nucleus and this cells. Look at the structure of female gametophytes. Female gametophyte. So this is the male reproductive parts and female reproductive parts. Male reproductive parts is the pollen grains. When fusion takes place, when pollination takes place, when cross pollination and self pollination takes place, what happens? Once you remember that, so datura flowers, what is pollination? Transforming of pollen grains from anther to the stigma. So this is the stigma, this is the pollen grains. When pollen grains transform from anther to the stigma, we can see some changes in stigma automatically. The stigma started releasing some sticky substances, some sugary substances. When pollen grains attach to the stigma, so this stigma started sticky substances and sugary substances. When stigma started release substances, sugary substances, this pollen grains converted into pollen tubes. Pollen grains converted into pollen tubes, which one having the 2 nucleus. This pollen tubes have 2 nucleus, first nucleus and 7 nucleus. 3 times mitotic cell division will take place in embryo sacs. So they will form like 7 cells and 8 nucleus in this embryo. Look at the exact structure of this female gametophyte cells. So it will divide like this. So in the embryo of female gametophytes, you can see the structures. So 1, 2, 3, 4, 5, 6, 7 cells is there inside the embryo. 7 cells. And they having 8 nuclei. 1, 2, 3, 4, 5, 6, 7, 8 nucleus. So it will divide like parts, like after once the division takes place in the embryo, these antipodals will move upwards. These are called as antipodals. Antipodals. In the middle of this embryo, we can see the polar nuclei. Polar nuclei. See the polar nuclei. These two are called as synergids. Synergids. This is called as egg cell. Egg cells. Egg cells. So what happens? Now come to the explanation. When pollen grain transforms from anther to the stigma, stigma started releasing sticky substances or sugary substances. When sugar substance is released in the stigma, this pollen grains converted into pollen tubes, which one having the 2 nucleus, first nucleus and second nucleus. These pollen tubes will enter in through this tile into the ovary through the microfile, when the small gap is there, into this embryo, female gametophytes embryo. The pollen tube will be entered into the ovary through the micropores. Fusion will take place in the ovary with the ovules. So here what happens? Double fertilization takes place in these flowers. What is this double fertilization? How this double fertilization will take place? See, actually there are 2 nuclei is present in the pollen tubes. The first nuclei will enter into the embryo sacs, is going to attach or fuse with the egg cells. First nuclei is going to fuse with the egg cell to form zygote. First nuclei is good. First pollen grains nuclei is going to fuse with the egg cells in the embryo sacs to form zygote. First fertilization is over. Then one more nuclei is present in the pollen tubes. This pollen tubes, this egg cell is already in the form of single N, haploid. So egg cell also having haploid, nuclei also having haploid, and egg cell also having haploid to form zygote to become diploid. 2N. Diploid is nothing but having 2 nucleus. Haploid is nothing but having 1 nucleus. One nucleus. When they fuse, when the fertilization takes place inside the embryo, fusion of first nuclei and the egg cell, zygote is formed. It is called as fertilization. Fertilization zygote is formed. And the second nuclei also present in the pollen tubes. It is also having single N. This second nuclei will enter in the microfile to the embryo, is going to fuse with the fusion of polar nuclei. This polar nuclei already have 2 nucleus. This polar nuclei already in the form of diploid. So this is going to fuse with the pollen tube single N to form 3N triplides. So this, when the second fertilization takes place in the embryo, fusion of second nuclei and the polar nuclei, triploid form These are very good food materials is present in this endometrium to the development of the seeds, and the fruits. So double fertilization takes place in this flower. First fertilization, first nuclei with the egg cell, second nuclei with the polar nuclei. It is called as double fertilization. So it's already having 7 cells and 8 nuclei. After the fertilization takes place, when the fruit is developing, this antipodals and the synergists will be disappears. They will be disappears. The fruit will be prepared. This oval will be converted into seeds. Oval will be converted into seeds. Like in beans, you can see the endosperms will be converted. They have seeds. But in like maize, like in corns, so the endosperms, they have a different seeds. That is the difference between the some like red grams and the maize and corn is a difference you can see. So they have cotyledons also. This is about structure of the ovals. Structure of the ovals, you can see pollen grains can attach to the stigma converted into pollen tubes having 2 nucleus. So mitotic division will take place in this embryo. 7 cells and 8 nucleus are formed. Once this pollen tubes entering into the microfile into this embryo, so double fertilization will take place inside this flower. First nuclei fuses with egg cells, second nuclei fuses with the polar nuclei. First one will be converted into zygote, second one will be converted into endosperms. This is about structure of the ovule. It is also very important for 4 marks. Next, the last one is seed germination. The last one is seed germination. How the seed will be germinate. How the seed will be germinate. We know that is a mature plant is there. I think few years back they asked this diagram of this seed germination directly for us. There is no matter, so you have to draw this diagram directly. You can see there are 2 different parts in this flowering plants. Mature plants. This mature plant started bearing flowers. Started bearing flowers. They have ovary, everything is there. So what happens here? Pollination will takes place when flowers started growing. It is called as fertilization. Fertilization takes place. Once the fertilization takes place, what happens? What is fertilization? Fusion of male gametes and female gametes. Once the fertilization takes place, zygote form. This zygote will develop into embryo. This zygote develop into embryo. This embryo will be developed into simple fruit. Zygote develop into embryo, embryo will develop into simple fruits. In the fruits, you can see some seeds is there. Seeds. So again, these seeds when you sow in the soil, what happens? They started germination. Germinations. So after the seeds, seedling. You can see seedling. The roots will go inside the soils, and you can see here the stem will grow into the atmosphere. Again, this seedling will become mature plants. This is about seed germination. last few years, 2-3 years back, directly this diagram. You can draw this diagram directly. Mature plants, pollination will take place, that is called as fertilization, fusion of male and female gametes. Zygote form, zygote will convert into embryo. Embryo is nothing but a fruit. In the fruit, seeds is there. You take colored seeds. Again, if you sow into the soil, they started germination, seedling, again mature plant. This is the cycle of this seed germinations. So today homework, structure of the oval, diagram and explanations, very important. Don't forget structure of the oval and diagram. Only draw female gametophyte diagram. And the next one is seed germination diagram. This cycle you have to draw. It's very important. Okay? Don't forget. Thank you, students.